Welcome to the BioInspired Institute at Syracuse University. My name is Jay Henderson, and I'm a professor in our Department of Biomedical and Chemical Engineering. Today we're going to share with you some projects in which polymers are being used to better understand human health and to design new strategies with which we can treat injury and disease. Behind me are the facilities of the BioInspired Institute, and we have many different labs working within a shared space. In those labs are polymer chemists, biologists, and engineers, among others, all working together to tackle tough problems that are very hard to pursue if you only have a single disciplinary perspective.
It's amazing. Mainly because I get to meet all these people that have different backgrounds and different goals. And what that helps with is I'm able to ask questions and kind of look at my work from different perspectives. So that has been like the most amazing part of getting to work in the BioInspired Institute.
Basically, we share one goal is to make the bioscience much safer and much convenient for all of us. So with this goal, we're actually creating this kind of substrate.
One of those labs is the lab of Dr. Beth Monroe, who will tell you about some of her work with smart polymers.
Hi, I'm Beth Monroe. I'm an assistant professor in biomedical and chemical engineering at Syracuse University. I run a research lab where we make materials to help with wound healing. One of our main projects is to stop bleeding in traumatic wounds like gunshot wounds. Approximately 1.5 million people die every year just from bleeding alone, and a lot of these people, including soldiers on the battlefield, die before they can get safely transported to the hospital. And then those that make it to the hospital often develop infections in the wounds that they received. And so our lab works to make materials that can go into those traumatic wounds like a gunshot wound and stop bleeding quickly so that they can get to the hospital safely. So ideally, we can continue testing these materials and ultimately provide a foam that you could put into your first aid kit and have available to use in case you have a bleed.
Another lab in the Institute is the lab of Dr. Zena Sze, who works with smart polymers and human-induced pluripotent stem cells to understand how cells and tissues develop and how diseases sometimes occur.
I'm Zena Sze, assistant professor at the Biomedical and Chemical Engineering department. My lab primarily works on stem cell technologies using a very special stem cell type called induced pluripotent stem cells. They are from human, and we use them to differentiate into cardiomyocytes, and then we use these cells as a primary cell source to engineer different model systems for better understand heart disease and also cardiotoxicity to different drugs. So I'm running a lab with 2 postdocs and 6 PhD students. We're going to primarily talk about the collaborative effort with Dr. Henderson to using their polymer system to engineer our heart cells to better understand in terms of some fundamental biology of the cardiomyocytes, basically the heart muscle cells, and also how we can use these heart muscle cells and also integrate with their polymer engineer techniques to better understand heart disease. My research direction has been changed so dramatically. So I think a lot of benefits actually came from the close collaborations and interactions at the SBI. So because like all of our lab is in this shared facilities, and we can always interact each other and understand what each lab is doing.
I use a special material that is called shape memory polymers, which can change the shape when they are experiencing some specific triggers, such as the elevation of temperature or light or electricity. And in this way, after they receive this trigger, the shape of this material will change, and we see the cardiomyocytes on it, and we study how those cardiomyocytes deal with this kind of active force that is bringing by this smart materials, and this is a base of understanding how those heart cells in our human body, how they deal with the daily change of their environment.
Also in the Institute is Dr. Pranav Soman, who is a biomanufacturer and works with optical printing to create complex materials and cell material constructs for areas like tissue engineering and organ replacement.
My name is Pranav Soman. I'm an associate professor of biomedical and chemical engineering at Syracuse. I'm going to talk about some projects in my lab and hopefully get you excited about doing science and going to STEM fields. My lab is primarily interested in advanced manufacturing approaches, and we get a lot of inspiration from what we see around in nature. So if you look around anywhere, essentially, you see a lot of really cool and smart things, right? So if you look at, let's say, butterfly wings, right? They have these really fantastic colors like blue wings and red colors and things like that. And one would imagine, when I was young, I used to think that these are just because of some kind of a color dye in their wings. But now I know that if I zoom in on this butterfly wing, I actually could see some micro scale structures. So these small scale structures are about 100 times smaller than the width of your hair. We get inspired from these kind of things in our lab to see if we can make similar things and sort of make advanced optics. And can we sort of process these sugar-based materials or protein-based materials which are found everywhere and shape them into complicated shapes and basically convert these simple material into smart optics, right? So one area of interest in our lab is called bioprinting, right? A lot of labs have started to use this kind of technology or extrusion-based 3D printing into extrusion-based bioprinting, right? So there the idea is that we replace the plastic spool or the plastic with a cocktail or a bio ink, which is basically a solution of hydrogel mixed with cells, and you extrude that out and then you print it on 3D on a stage and you make it into a 3D construct. But now this is a living entity, right? The cells inside are stuck in the gel, but they are actually happy and alive.
And this is our printer that can scale between multiple scales to achieve resolution down to 50 micron all the way up to centimeter size. The main differences between printing. So with extrusion based, that technology is something that is typically used for large scale stuff, rapid prototyping because it's very cheap, it's relatively quick in speed to do, and it's somewhat easy to do as well. I mean, you buy a printer off the shelf, download some software, and upload and go from there. But with our printer, it's light-based, so it's a little bit different. It uses light instead of a solid material, just extruding. Extrusion-based takes the layers and determines points. So at each layer, it's going to determine, thousands of points to lay a bead in the correct pattern. With, switching over to light-based, you can imagine light-based as being like a UV lamp. You're turning like a flashlight on and off. We are cross-linking, which is basically solidifying a liquid to a solid. So instead of a hard plastic extruding out of a nozzle, we're using a liquid bath, and with the light, that turns something solid.
One of the projects in the lab is basically focused on this problem, which would allow us to scale bioprinting not only to small-scale organs, but large-scale organs, right? So you have an active supply of food and oxygen and removal of waste. So we build this entire construct, which is pretty large, but as we build it, we also build plumbing or pipes which are embedded inside the construct, so that once the whole thing is printed, we can attach some kind of tubes to the inlet and outlet of this construct and start to flow in media or food and things like that to ensure that this bioprinted construct stays alive.
That's a HLP, is hybrid laser printer. So it's very, very powerful in printing very, very small structures. For example, you can print a structure with the smallest feature size of 200 nanometer. So we are mostly using this printer for doing, tissue construct. So one of the inability of current science, I would say, is to incorporate blood vessels in tissue construct. Maybe you want to make let's say, a bone construct, and you want to put that in a patient. But without a vasculature there, without a blood vessels there, that's going to fail, right? So basically, what we are doing is we kind of like, let's say, print bone with the help of your additive part. It's quite exciting as well as it's challenging also.
Understanding how cells interact with the complexities of the environment is important in tissue engineering, as well as various therapeutic applications. So my work is aimed at trying to pioneer a way that we can capture this complexity so that if we wanted to create a model of a disease, we can utilize the process that I'm working on.
Thanks very much for visiting us today at the BioInspired Institute at Syracuse University. I hope that this has helped you better understand how polymers are helping us do amazing things that hopefully will impact human health in the very near future. Thanks for visiting.